My name is Ong and I'll be explaining about my final year project in this video. For this project under Prof. Jagger's guidance, we set out to focus on Parkinson's disease classification through multiview learning approaches. The study of the human brain is a fascinating area. The brain is made up of 100 billion neurons and is larger than expected for our body amongst mammals, taking up to 20% of our body's energy budget. Due to the complexity, we've yet to come to a full understanding of it. Neurodegenerative diseases such as Alzheimer's disease and Parkinson's disease are becoming more prevalent in the recent years and are the top causes of dementia for the elderly. Detection of these diseases prior to memory decline symptoms is possible through brain scans. Machine learning models are able to analyze those brain images and predict the progression of the disease. With the complexity of our brains, there are various ways of brain imaging, and they can be divided into two categories, structural and functional. Under structural imaging, we have methods such as MRI and TTI, and these are meant to inspect the gray and white matter of the brain. Functional imaging methods include EEG, PET, and fMRI, which are more concerned with measuring the activation of the brain through oxygen use or blood flow. These brain imaging datasets also usually have high dimensionality. Instead of using machine learning models on one imaging modality, we could combine the information from both imaging modalities into a shared representation of lower dimension. Previous approaches to machine learning were based on canonical correlation analysis, which projects each view such that the correlation between these projected views are maximized. It is further extended by GCCA, which allowed multi-view learning for more than two views. Its extension DGCCA is able to also capture more complex relationships between views. DGCCA will be more suitable for our use case due to the complexity of our dataset. In this experiment, we use DGCCA to combine different DTI metrics for Alzheimer's disease detection by using feed-forward neural networks. We perform classification between AD progression class by S, and if we focus on the last two columns, we can see that DGCCA fuse representation performs consistently better than simply concatenating the features, although it can be sometimes driven by one DTI metric. It should also be noted that we are now able to perform classification on a much smaller dimension data. When we tried to combine structural and functional images for AD patients, we did not have enough patients that went through both, which will give us very little data to train on. For the next experiment, we resorted to classification between Parkinson's disease patients and the control group by combining TTI and fMRI scans. Let's have a look back at the DGCCA architecture. Here, the DGCCA architecture contains a feed-forward neural network, which transforms an input data to a specified dimension, and we learn a shared representation G on it. The brain connectome data that we are using is a connectivity matrix, and when we use DGCCA, we are forced to flatten the inputs to fit into the feed-forward neural network, causing information to be lost. Therefore, we propose a modification to DGCCA by using graph convolutional networks instead of the feed-forward neural networks, and it will better encode the connectome data. These are the results for PD classification through DGCCA and GGCCA, compared to GCCA and just using one view. We can see that all the multi-view learning approaches did not manage to do better than just using one view. When using GCCA, the models did not manage to learn any useful information from the fuse representation. The poor performance of our models on the PD data could be due to limited samples that we have. For us to combine different views, one patient needs to go through multiple brain scans around the same time range. The dataset is also extremely imbalanced with the PD class making up to 90% of the entire dataset, and therefore we have to remove some of the majority class for classification. The input is also of very high dimension, and we need more data to train the additional weights introduced by the DGCCA and GCCA architectures. Therefore, in the future, we could be trying out other datasets with multiple views, a graph structure, and that contains sufficient number of samples. I have summarized my FYP project in this video, and thanks for watching till the end. I'd like to take this opportunity to thank Prof. Jagger for supervising me and Ehao for mentoring and guiding me throughout the whole project.